Welcome back. Synthetic biology has been a recognised field for about 10 years, and acquiring the skills to develop a terminator circuit that can kill cancer cells doesn't seem like bad progress to me. Nevertheless, some of the fanfare and hype that surrounded synthetic biology 5 years ago is gone. And there's a simple reason for this. The tools outpaced our knowledge. The cost of genetic sequencing and synthesis continues to plunge, but the functions of many genes and even the simplest forms of life, like bacteria and yeast, remain elusive. Genetic networks interact in complex, mysterious ways so that engineered parts do unexpected things when inserted into genomes. That's because cells are machines whose complexity is many orders of magnitude greater than a man-made engine or computer. We sometimes call the unpredictable variation that results from the complexity of life noise. Noise is a combination of all the things we can't quite account for. Things like natural variation in the behaviours of genes or proteins or molecular interactions that haven't yet been described. Synthetic biology has meant confronting noise and scrutinising it. That's leading to a lot of new knowledge on cell biology. That Richard Feynman quote that to understand something you must first be able to build it is big in synthetic biology circles. We'll know that we understand cell biology when we can build a cell from scratch. But then you'll still have evolution elbowing its way in. The DNA you put into your cells will mutate. And these mutations will be selected for or against. And if they are selected for, then they will accumulate in your genetic circuits and they'll change their function. Evolution is a phenomenon that would drive any electrical engineer mad. Well, despite all that, the field is moving in a positive direction. It's just that the road is far longer than some imagined. And this isn't surprising. Gartner, an information technology research and advisory firm, has coined the term hype cycle to describe exactly this sort of phenomena. A new technology is introduced with enthusiasm and it will be followed by an emotional low when it fails immediately to live up to its promise. But Gartner also discovered that the cycle doesn't typically end in what the firm calls the trough of disillusionment. Rising from the ashes is the slope of enlightenment, meaning that when viewed from a longer-term perspective, the majority of these much-hyped, groundbreaking developments do eventually break much new ground. And this is what is now happening in a lot of new genetic areas like personalized medicine and synthetic biology. So, for example, because DNA design, synthesis and assembly are now so much easier, many are talking about moving away from tweaking existing genetic designs to the construction of whole new organisms, species birthed entirely from our imaginations and capable of feats impossible in the natural world. You've probably heard of a certain scientist called Craig Venter. Since the 1990s, Venter has wanted to know what the minimum genome required for life would look like and what it would take to write a genome. But back then, DNA synthesis technology was too crude and expensive for anyone to think about writing a genome, while recombinant DNA techniques using restriction enzymes and ligases to cut up existing DNA and stitch it back together again were too unwieldy for the task. Huge advances in biotechnology mean that this work has moved from the molecular to the digital. DNA is information technology. Genetic code, the written sequence of A's and T's and C's and G's, is cut and pasted on the computer. DNA bases and sequences can be swapped in and out with precision, and then, when the code looks right, simply hit send. A dozen different commercial DNA print shops can now turn the digital code into biology. In 2010, Venter announced that he had created the world's first synthetic life form, Synthia. Synthia was a cell whose 1 million base pair genetic code, copied and modified from an existing bacterium, had been assembled not by its parent, but by a computer in Venter's office. Once the code was complete, it was emailed to Blue Heaven Biotechnology, a company with a lot of DNA synthesizers, and that sequence, including literary quotations and website addresses, was then jammed into another similar cell which didn't have any DNA, and the whole thing booted up. It did live, and it hadn't lived before. It metabolized, grew and divided. The image behind me shows a bunch of Synthia cells dividing. Well, for their purposes, Venter and his team define a synthetic cell as one that operates off of a chemically synthesized genome. But it doesn't fall under the tight definition of synthetic biology according to the purists, because the organism was still mostly a copy of one in nature, and to be a designer life form, it would need to have its genetic function changed in some deliberate and more useful way than just having literary quotations. What Venter and his team did was an exquisite piece of genetic synthesis. Now, Craig Venter is incredibly good at generating headlines and PR. He also gets a lot of stick for these two things. But he's a revolutionary, and Synthia is a really important technical achievement. It shows we're not just capable of stringing a couple of genes together, but also manufacturing an entire genome. Even if it's only the smallest bacterial one we know of, it still shows just how good we are at synthesizing DNA. And this is highly important. The old era belonged to DNA sequencing, reading the strings of A's and T's and G's and C's in DNA. Writing DNA creates many more opportunities. Synthia is just the beginning. Teams worldwide are currently making 16 synthetic chromosomes to create a synthetic yeast. Synthia is a bacterial cell and therefore has no nucleus. Like humans and plants, yeast is a eukaryotic organism that contains complex structures that store DNA within a nucleus. Yeast was picked as it only has about 6,000 genes, which makes it small compared to other more complex organisms such as plants. And following the Synthia model, hunks of DNA will be designed by teams on computers, which will then be synthesized by specialized companies. The team will then assemble the pieces of DNA in a sequential manner and insert them into a yeast cell, first stripping out its natural DNA. Well, Venter's grand scheme for Synthia and the Minimal Genome project he's been working on is to create a living template on which you can add functions. And that really is a synthetic biology project. As Venter says, it's like creating a basic chassis for a car, which is then available to decorate with whatever you want. Likewise, once complete, new strains of synthetic yeast could help make products such as vaccines, biofuels, and chemicals. So synthetic biology, though, is not just about medical and pharmaceutical applications. It goes a lot wider. You can also think about it in the context of manufacturing. As engineers, synthetic biologists look on biology not just as a science, but as a manufacturing partner. And what Venter is really interested in dealing with is energy production and environmental issues. Venter is interested in generating custom bacteria, designer bacteria, that will be provided with the genes to generate fuel, plastics, clean up toxic waste, or provide food. The appeal of these designer bacteria is manifold. They will reproduce themselves and thereby generate not only their product, but more biological factories. And designer bacteria could produce the same product as an industrial plant while feeding on pollution or sunlight. Photosynthesis is powered up by 90 terawatts of energy all across the planet. We can't use all that energy very well right now, but we can imagine partnering with biology to make the things we need without putting such an environmental load on the planet. So Venter and his group aspire to replace the petrochemical industry with strains of algae secreting high yields of fuel. They also think agriculture, as we know, needs to disappear. If strains of algae could secrete high yields of protein using less land and water than traditional crops, it could represent the best hope of feeding a booming planet. Well, to some extent, of course, this kind of engineering is already happening, albeit with less ambitious aims. As I mentioned, it's now routine to use microbes to make medicines, and at DuPont, engineers are using modified bacteria to produce polyester for carpet. That has been achieved already to show that we're only grazing the surface. I already mentioned the engineered yeast that produces the anti-malarial drug artemisia. Jay Kiesling and his team stitched together 10 synthetic genes derived from 3 different organisms to create this yeast strain. Well, of course, to create a flexible set of designer life forms, you need genes that can get the job, whatever it is, done. In 2003, Venter sailed around the world in his boat in search of new forms of microbial life with novel genes that might be useful in synthetic life. And to date, they've discovered about 60 million new genes that include some that help organisms survive in polluted waters, or that could be used to produce hydrogen or new antibiotics. Very useful though this is as a means of assessing the Earth's genetic diversity, this approach is no longer cutting edge for synthetic biology. Even the current set of biobricks may soon be redundant. The Florida-based Foundation for Applied Molecular Evolution has added 2 bases, Z and P, to DNA's traditional 4, so augmenting the old genetic alphabet, allowing proteins to be made with amino acids that don't yet occur in living things. In future, designer organisms could contain genes made from scratch, making completely new types of protein with functions never seen in nature. Well, this is going to require the ability to arrange nucleotides in the order required to produce a protein to a precise prescription. And thanks to supercomputers, protein chemists are increasingly able to predict how a given gene will fold and hence predict how the protein will behave. Well, all this means that the ultimate limits of synthetic biology are hard to discern. That's probably true of most genomic-based fields of study, not only because of the new technologies, but because there is this great movement promoting the free exchange of ideas, the free movement of technology, a democratization of science built into genomics and its offshoots. So there's unprecedented freedom of information and publication of genetic information. In fact, when researchers publish their work today, they upload the genes and the phenotypes and the genomes and transcriptomic profiles online in large databases. Well, the transcriptome, by the way, is the set of RNA molecules found in a specified population of cells. It tells you which part of the genome, the DNA, is actively expressing RNA. Well, these databases are available to researchers and the public for free. The United States National Institute of Health, National Center for Biotechnology Information, curates websites like GeneBank, which contains gene information on humans and other organisms, and the Gene Expression Omnibus, or GEO, which contains thousands of transcriptomic datasets describing when and how your genes turn on and off as determined by levels of messenger RNAs. The database of genotypes and phenotypes, or dbGaP, contains over 50,000 samples from studies which try to connect people's genotypes with their observed phenotypes. This data comes from experiments with SNP genotypes, transcriptomics, and sequencing results. They contain all of the demographic, clinical, and exposure information on the study participants. The European Bioinformatics Institute, or EBI, manages similar tools and databases online as well. Well, these databases are an invaluable resource for GWAS studies. Having all this information publicly available means that bright people all around the world have been able to rub shoulders and exchange ideas. That's massively accelerated progress and led us into many new creative directions and that not have taken place if only small numbers of people had been involved. As an example, the democratic process built into the BioBricks ethos, where everything is freely available and open access, is allowing a new generation to embrace its vision. Since 2003, every year, high school and college students compete in an international genetically engineered machine competition known as iGEM, where they need to think of a problem and design and build a solution using only BioBricks. Here's a high school team from the 2012 high school competition. Looks like they're having fun. These teams have pushed not only technical challenges, but creative ones as well. 2011's European champions designed a system for preventing soil erosion and the conversion of land into desert. The teams come from diverse backgrounds, maths, engineering, even astrophysics, so they are unfettered by the narrowly defined science disciplines.